So our next speaker is Peter Kulakow, and he is leader of the Cassava Improvement Program. And we heard yesterday quite a few times or over the last day and a half about corn and cassava, sort of the main crops for the African continent. So that's his major thrust. And he's works at the International Institute of Tropical Agriculture in Nigeria, and he's the cassava breeder and genetist. So that's what he has been doing, and we want to hear about it. But before, he received his PhD at University of California at Davis, and then he has worked at Land Institute in Salina in California, and at Landscape Ecology at University of Nebraska. So he's got a lot of experience in different fields, and in environmental engineering and technology at Kansas State University as assistant prof before he accepted and being at IITA. So he's going to talk about cassava, and as I mentioned earlier, we have legumes in India that's been the focus. Cassava is a major focus in Africa. So Peter, he's going to talk about advances in cassava crop improvements to benefit farmers in Sub-Saharan Africa.
Yeah, thank you very much. I guess that my background shows that I've been a part of multidisciplinary teams quite a bit, and this project is also very much a multidisciplinary effort, and it involves many partners in many different places. So I'll just highlight a few emerging technologies and how they are impacting cassava breeding in this particular situation. Let's see. So cassava is a perennial, clonally propagated tropical root crop. The cropping cycle is about 12 months. If you harvest it in less than 12 months, then you have to store the stems. Each part of the plant is important. The roots, of course, for starch production, the stems for propagation, and the leaves are also used for food consumption, feeding to livestock. Relatively small amount of the biomass is actually recycled from one cropping cycle to the next, which is quite important. Here are three cassava breeders, Hernan Ceballos, me, and Elizabeth Parks. This is actually the same genotype in two different situations, so you can see how plastic cassava is to the genotype, environment, and management situation. Cassava is important food security and income generating crop for about 500 million people. The research community is relatively small, historically with maybe little research investment, but that's not so true at the moment. There's been the statement that there are only 5 cassava breeders globally. There are really about 50 cassava breeders globally. There is a need to have expanded financial support to maintain a full cassava breeding pipeline. A lot of these breeders are in national programs that are very poorly funded in many different countries. In 2014, by FAO stats, there are 270 million metric tons of cassava produced, so that's 5.4 million metric tons per cassava breeder. If you look at yields, this is on 24.2 million hectares. So the top 5 African countries producing cassava, the average yield is 11 tons per hectare. The top 5 Asian producing countries, 24.9 million tons per hectare. So you see quite a big yield gap between Africa and Asian countries. Most cassava varieties have a yield potential of 30 tons per hectare or more. But there's, in addition to breeding though, there's many important allied disciplines that are also critically important to the cassava value chain. Cassava is important now, but it'll likely continue to be important, perhaps more important in the future. There's many different ways of looking at cassava suitability, but in general, even though the suitability in some areas may go down in the future, say in West Africa, it's still going to be a very important crop long into the future in Africa. Cassava is primarily a food crop, even though we often talk about it as an industrial crop, at least in Africa. In Nigeria, only about 5% is boil and eat, so that's fresh consumption. About 70% of it is gari, which are fried starch granules, and then about 15% is other products, including fufu and lafun and other products. These are all fermented products. to deal with the fact that cassava roots have cyanogenic glucosides, and so the cassava roots need to be processed in many situations to be safely consumed. About 10% of the production is industrial. Of course, there's a lot of marketing in all of this, but the industrial production, there's flour, starch, ethanol. Cassava bread is an important impetus to enhancing demand for cassava flour. So this is an area that's received a lot of attention. There are many development challenges that require a full value chain approach to address cassava breeding and all of cassava development. Of course, there's land degradation and important soil erosion, reduced soil fertility, climate change. Diseases and pests are critically important, especially drought and virus diseases in Africa. Limited labor, there's an aging farmer population and the need to attract youth to agriculture. Mechanization is very low in Africa, and there's need to improve mechanization both in production and processing. Everybody needs access to quality seed, fertilizers, and other inputs. There needs to be access to markets and market information. And you need to deal with the poor infrastructure, especially transportation, power, and water. So those are all realities that we deal with in Africa. So I just want to highlight 3 cassava breeding-related projects that are part of the recent and very generous donor interest in cassava in Africa. And the first is the NextGen Cassava project led by Cornell University to enhance genetic gain in cassava. And when we talk about genetic gain, ultimately we need to be talking about performance in farmers' fields, not just in experimental plots. This is funded by Bill and Melinda Gates Foundation and UK Aid. There's also Harvest Plus to improve the nutritional value of cassava through biofortification with provitamin A. And then a very new project that's just starting, building an economically sustainable cassava seed system in Nigeria. And that's led by RTB. So the NextGen Cassava project started in 2012, and it's a collaboration of a number of different organizations. It's primarily designed to test genomic selection in Nigeria and Uganda. These are the Nigeria and Uganda national programs. Also looking at flowering in cassava, developing a globally accessible cassava breeding database called cassavabase, enhancing germplasm exchange between Latin America and Africa, improving the cassava genome, a special initiative to improve gender-responsive breeding, and capacity development for students in national programs in Uganda and Nigeria. I'll start with the gender-responsive breeding because it's important in cassava that women are a very important part of the cassava system. Matter of fact, in many areas, they're both the farmers and the processors of cassava, but often they're not listened to sufficiently in developing priorities for cassava. So we have an initiative to help understand the drivers for cassava variety adoption, both for men and for women, as well as youth, because the different groups have different needs. And we want to respond to sex disaggregated trait differences in breeding our cassava into the future, use this to prioritize traits. We want to improve our phenotyping methods and selection, especially for gender-based traits where they are important. And we want to introduce more gender-responsive participatory evaluation. In the end, to have varieties that will be readily adopted and wanted by the farmers that produce and use cassava. This is just a few examples from some sex disaggregated ranking of cassava traits in 6 villages in Nigeria. High yield is important for both men and for women. And in fact, women mention yield more often than men do because they focus on root size as well as just overall production. This is partly because of the importance of having cassava roots that are easy to peel. Dry matter content is also readily mentioned by farmers, although they usually talk about it as cassava roots that have too much water. And the other trait that farmers often mention that's not adequately been addressed in breeding programs is maturity time. They want early maturing cassava, and cassava that has variable harvest options. In general, for women, though, it's important to have access to planting materials and access to information that has often been limited in the past. So the cassava breeding cycle takes a long period of time because of the low multiplication rate of cassava stems in order to have multi-location replicated yield trials. So one cassava breeding cycle could take 5 or 6 years with adequate phenotyping. But with genomic selection and using genotypes to predict phenotypes, you could have an annual breeding cycle. And this is what we are trying to test in looking at genomic selection. So genomic selection involves developing models, having a training population where you have both phenotypic and genotypic data to develop a model to estimate breeding values of lines that can be selected and advanced into a crossing program and run a relatively rapid cycles of selection using this relationship between genotype and phenotype that's established in your training population. At IITA, we have a historical collection of what we call genetic gain that has over 700 genotypes, and we have used this, it's been evaluated in over 45 environments, so we're using this to develop our training population. So at IITA, we're testing an annual recombination cycle where we go from seed to seed in a single year. At NRCRI and NACRI, the national programs, they're using a 2-year genomic selection breeding cycle. So far at IITA, we've actually selected our 3rd cycle of selection. So this is the initial training population, 1st cycle and 2nd cycle. Our training population now, it started out with 969 genotypes. Now it has, I guess I'm losing the battery on this. It now has over 5,000 genotypes in it. And what we've seen in terms of the first 2 cycles of selection for highly heritable traits like dry matter content and CMD resistance, we're seeing a response to selection. You see the genomic estimated breeding values are improving from cycle 0 to cycle 1 to cycle 2 on the left-hand side there. On the right, a low heritability trait like fresh fruit yield, we did not see a response in the 1st cycle. It looks like we're starting to see a response in the 2nd cycle of selection. We have proper response to selection trials underway now that will look at this in much more detail. By the end of the 1st phase of the project, we want to have 4 cycles of selection, which is pretty good. Now, one of the things we've done in the NextGen Cassava project, though, is to make all of our data completely publicly accessible through cassavabase. So you can actually go look up all of our trial data from the past years on cassavabase. It's cassavabase.org. It's fairly easy to use. There are over 80,000 accessions in it so far, over 300,000 trial plots, and over 8 million phenotypic observations recorded there. Over 2 billion genotypic data points, and there's 360 registered users of cassavabase so far. It has many different breeding management tools. This is just pedigree management, and you can see what genotypes are derived from what, or in a lot of visualization tools. We also have a barcode system for helping to track genotypes. There's many other tools, trials management, crossing management, field book tool, the genomic selection tool, genetic maps, and statistical tools. In particular, the field book, which is initially has been done on electronic tablets, is now being done on smartphones. So a lot of our breeding is actually done directly on the phone. So that's a very promising development, and it also allows us to get our data into a situation where we can analyze it very quickly. There are a number of initiatives to improve phenotyping of cassava, and this is a place where we also invite more collaboration. The field book is, of course, has been very useful for just normal breeding traits so far, but there's the 1K tool is part of a tool being developed with Kansas State University and other partners as part of one NSF Bread project to look at capturing images and analyzing images directly using smartphones. This is an example of images of cassava roots in a clonal evaluation, and you can very quickly look at the size, volume, and shape of roots that can be readily photographed in the field. So this is turning out to be very useful, and this will be very important for developing cassava genotypes that are suitable for mechanization. We're also working on developing ground-penetrating radar. This is a project led by Texas A&M, and also CIAT is a major collaborator on that. We're very interested in using it in Africa. It's important for non-destructive estimation of root development, which is something that's been very difficult to track without destructive and very expensive harvest operations. So the ground-penetrating radar instruments are even much smaller than this in this picture now. So that's something that's developing quite nicely, and we look forward to that in the near future. If you look at genomics, there's been a lot of advances. We're up to the 6th version of the cassava genome. The first version was published in 2009. It looks like we're going to stop at about version 7, which is in production right now. But this picture shows the paleotetraploidy event that happened in cassava. You can see each little piece there represents one of the 18 cassava chromosomes. About more than 30 million years ago, before rubber, Hevea and Manioc diverged, there was a paleotetraploidy event that can be seen in analysis of the cassava genome. It just shows some of the power of the tools of genome sequencing that we're able to harness, as well as to characterize the genome much better than we've ever understood before. This is all summarized in a Nature Biotechnology paper that was published this year. Each line of this chart is a different genotype that has been subjected to genotyping by sequencing, and where we have over 40,000 SNP markers. And you can see most of that orange there is Manioc germplasm, but the green are introgress segments from wild cultivated crosses that were done in Tanzania as part of an effort to introduce cassava mosaic disease resistance into the cassava germplasm. So you see on chromosome 1 and chromosome 4 this history of introgression of wild germplasm into the African cassava varieties. That large section called TMS in the middle represents a lot of the genetic gain population and the current important breeding lines of cassava in Africa. A little bit more on cassava mosaic disease. It's the most important cassava virus, although there's another very important one as well. There's actually 2 species that are important, African mosaic virus and East African mosaic virus. You can see they have varying levels of symptoms, and it's primarily been managed by resistance, both quantitative and qualitative types of resistance. You can see in the center there a resistant variety and a susceptible one on the right, and how it impacts root production. But one of the things we've learned through the genotyping is that we have a very narrow base of cassava mosaic disease resistance that's primarily located on chromosome 8, as summarized in this paper in Plant Genome. There are other parts of the cassava genome that contribute to resistance, and we need to capitalize on that. That's more of the quantitative resistance. So through that, we've learned that cassava mosaic disease resistance, although it seems to be durable, it's very narrow, and that's a concern. There is some evidence within this narrow segment that there might be more than one locus involved, or perhaps some multiple resistance alleles. So we need to understand that chromosome segment much better. But it does allow us, with genomic predictions, to understand the inheritance and our ability to select for cassava mosaic disease resistance. This is an initiative in Hawaii where we're hybridizing Latin American and African cassava. We're doing it in Hawaii because the Latin American cassava is completely susceptible to CMD and is very difficult to hybridize in Africa. And it's impossible to send African germplasm to Latin America because they don't want CMD. So what we have done is found a neutral place to do hybridization, and this promises to be very important in the future for transferring germplasm between Latin America and Africa. So I just want to show you a little bit here about biofortification of cassava. The sample on the lower right here is the best biofortified cassava variety in Nigeria. It has 12 micrograms per gram fresh weight of total carotenoids. You can see a white genotype over here on the left in terms of comparison. So this best biofortified variety we're very proud of it. It's very good. But you can see we've achieved a breakthrough in a seedling nursery that we harvested in April of 2016, where we have a large number of genotypes now that are above 20 micrograms per gram fresh weight of total carotenoids. And this should translate to more than the target level of 15 micrograms per gram fresh weight for beta-carotene, since most of the carotenoids in cassava do happen to be beta-carotene. We're able to do this using a technology developed by the private sector in Germany, the company BioAnalyt. They developed this device called iCheck Carotene, it's a portable spectrophotometer that we can, it's battery powered, and we can take it to the field, and we can get results from our germplasm screening within the same day, and that allows us to make selections very quickly. If we go through a conventional spectrophotometer or HPLC, it takes much longer for us to get results, and it's very difficult for us to make selections. So these kinds of tools, that low-cost phenotyping tools, are really important to fast-tracking our breeding program. Okay, thank you very much. Yeah. There have been 6 biofortified cassava varieties released so far in Nigeria, and by this year they will have reached over 1 million farmers. Harvest Plus has a very effective delivery process to catalyze the distribution of improved cassava varieties, and this just shows some pictures from a nutritious food fair that was held in Abuja in 2015, in November of 2015. And finally, I just want to talk a little bit about how you identify cassava varieties and what that means for cassava variety adoption. Using genotyping by sequencing, we established that we could use DNA fingerprinting to identify cassava clones in farmers' fields. This is an example of 500 households in 3 regions of Ghana. Each vertical strip here includes one variety assay. So we have over 700 different varieties that were collected in this particular collection of genotypes here. And you can see that there's 11 large clonal groups that account for over 2/3 of the cassava varieties that were identified in these 500 households in Ghana, including this very large orange group 1 on the right, showing that it's a very large a clone that's very important in Ghana. But the idea of this and the ancestry analysis that you can do with genotyping by sequencing allows us to be very accurate about identifying cassava varieties. Prior to this, it's just been expert opinion about what a variety is and farmer names. And in particular, you see here, this is the farmer names on the left and their relative importance in terms of the size of the letters. The 3 most common farmer varieties in Ghana are actually all the same clone. And we didn't know this prior to that. So it's very important information. Some names are multi-purpose. They refer to many different genotypes. Other times you have many names just referring to the same genotype, which is common. But anyway, genotyping really is it's a really game changer in allowing us to understand farmers' use of varieties. And one of the things in a larger study in Nigeria that's still being analyzed, we've really come to find out that farmers are not using improve varieties more, mostly because of lack of access to planting material. It's not so much that the varieties don't have the characteristics farmers want. And when you see disadoption, it's really local varieties that are adopted more than improved varieties. That seems to be a conclusion that we need to look into this more, but it's a little bit, but it's promising in terms of the potential for improved varieties. But we need to get those varieties out to farmers. And this brings me to just the last project I want to highlight that's just starting. It's a collaboration of many organizations led by the Roots, Tubers, and Bananas CGIAR research program, and it's to develop a sustainable cassava seed value chain that's characterized by commercial production and dissemination of high-quality cassava planting material. And we have a number of different models to test how to produce and move cassava through the seed production value chain from pre-basic seed, basic seed, and then various types of commercial seed. We have a model where we're working directly with commercial cassava processors to produce their own seed. And then we also have small seed entrepreneurs that are more of a village level seed production system. But this is very important because there needs to be some kind of a system to adequately produce planting material and get it out to farmers. And this is quite a challenge for clonal crops, so there's no underestimation of the challenge here, but it's with the support of the Bill and Melinda Gates Foundation, we're working on this great opportunity. One of the final technologies that we're testing in this effort is it's called SAH, semi-autotrophic hydroponics, and it's a system that was developed for potatoes to rapidly multiply disease-free potato clones. But we've done a pilot test, and it looks like it's very promising for cassava, and it could be a game changer in terms of our ability to scale up varieties from very small numbers to very large numbers that are needed for effective seed markets. So finally, just in conclusion, the next generation breeding really offers a combination of technologies, including genomic selection, to increase rates of genetic gain in cassava. Cassava breeding data is freely and globally accessible to all scientists, and we invite your contributions to be part of the cassava breeding team. Biofortification of cassava will be able to meet its goal of 15 micrograms per gram fresh weight, and offers communities improved nutrition options. And then commercial seed systems do promise to improve farmer access to improved varieties. Thank you.